This is part 3 of Chapter 4, and it actually is going to be covering what is found in Section 4.3. So we were talking about how the basic structure of male and female hormones is really identical, or at least almost identical. It has the same carbon skeleton with the different rings, the 4-ring structure. But the only difference is the attachment of different functional groups at 2 places. So the difference between estradiol and testosterone is very, very simple, and it makes a world of difference. And I ended up being cut off there when saying the difference between male and female in terms of hormonal differences, but in terms, of course, of genetics, we go back to our X and Y genes. So on to 4.3. 4.3 is a discussion of functional groups. So those differences between estradiol and testosterone are what is attached to the carbon rings of the steroids. And those attachments are primarily going to be functional groups. So functional groups are the common parts of organic molecules that actually are involved in chemical reactions and really do a lot of determining how a different biomolecule is going to react and interact. So the number and arrangement of these molecules actually give it distinctive properties because the different functional groups can have different electronegativity. They can be hydrophobic or hydrophilic. They can be stable. They can be ionic. So there's all sorts of things. They can be polar molecules. So all of those are going to play into how the actual molecule they are attached to acts within the body. So it affects what we call the reactivity, making hydrocarbons, which are very stable and non-reactive, making the actual molecule as a whole more reactive, and in some cases even hydrophilic instead of being hydrophobic hydrocarbons. So the main functional groups that you need to know are listed here, and we're going to go through each of them really quickly because really it's going to come down to just memorizing them, and really what you need to know is the name of it and its basic properties. So is it going to be polar? Is it going to have electronegativity? Is it going to make it hydrophobic or hydrophilic? Those types of things are what you should know about these groups, along with their names. So the first one is the hydroxyl. So the organic compounds with the OH end up being mostly alcohols, and they typically end in an OL in terms of their name, like ethanol. They make the compound that they're attached to polar and hydrophilic. And that comes down to the fact that they have this oxygen attached. So when you get an oxygen attached in the functional group, you're changing it to a polar molecule in most cases, and something that's hydrophilic. Because as you know from water's properties, oxygen is very electronegative and it is very hydrophilic. The next group is a carbonyl. That's a carbon that's double bonded to an oxygen. So a carbonyl is at the end of a molecule. It's called an aldehyde. When it's in the middle of a molecule, it's called a ketone. So you can see here how that might look with these two. I know this is really small, but this one's propanol, and you can see it's an aldehyde, and it has the CO, the C with a double bonded O, at the end of the molecule. And here we have it found right in the middle of the molecule, so this is actually classified as a ketone. And again, this is not going to be quite as electronegative because the oxygen here has got this double bond here with the carbon, but it still is going to make this more reactive than the hydrocarbons typically would. The next one sounds like carbonyl, but it's actually carboxyl. And so in this case, we still have our carbon with its double bond to oxygen, but there's an additional OH attached here. So we have a CO and then an OH. So C double bonds to O and then single bonds to the OH group. So that it would look something like this. These end up making weak acids. They are making up fatty acids. They're also one of the major components of an amino acid. So this functional group you will see a lot of. The other functional group that's on an amino acid is, not surprisingly, called an amino group. So the NH2 is an amino group, and it looks like this. The NH2 acts as a base because it actually can pick up hydrogen from solutions. So this NH2 can become NH3 very easily. So that's why this particular group will act as a base. So it gives basic qualities to amino acids. So if you look over here, we have an electronegative end to an amino acid, and then we have a the other end that's actually acting as a base. So that gives us interesting properties with our proteins. So this next group has a sulfur and hydrogen bonded to the carbon backbone, and these make up thiols. And these groups stabilize the structure of proteins because this actually makes a very stable, non-reactive group. The phosphate group you'll see a lot of as well because it's a big part of our nucleic acids. So when we have phosphate binding with 4 oxygens, that's called a phosphate functional group. So it's PO4. The O, lots of oxygens there, means lots of electronegativity, lots of negative charge. So it's highly reactive. We also see the phosphate group in something that's really important to life, and that's ATP and ADP. So those molecules that are the carriers and the storage units for energy are holding phosphate, and that is what gives them so much potential energy, is all this electronegativity with these oxygens surrounding the phosphate. Okay, so the next group, methyl. CH3 is called a methyl functional group. That's just carbon bound to 3 hydrogens. It acts as a marker for molecules, and when it's added to DNA, it can actually affect the expression of genes. And so when we actually look at DNA in the unit that we're going to spend with molecular biology, you're going to see that methylation of DNA is really important with gene expression and determining which genes are actually being transcribed and expressed. Okay, so the last thing that we are going to look at with this lecture is ATP. So we mentioned that before with these phosphate groups. ATP stands for adenosine triphosphate. So not only do we have one phosphate group attached to the adenosine, but with ATP we end up with 3 of these. So again, we're packing a lot of potential energy, and there's the potential to react, to release energy. And that's how we get the energy that we need for the cell's processes. And when we just remove one of these phosphate groups, we go from ATP to ADP, which is adenosine diphosphate. So you can see that process here with ATP losing one of its phosphates. When it reacts with water, we get a release of energy and the formation of ADP. Very important process that we'll revisit in a lot of our units. So that's the end of Chapter 4. And I'm actually going to go ahead. We still have a few minutes left on this lecture, this tape. And so I'm going to start with the next unit, which is Chapter 5. Because as we did in lab this week, you're building macromolecules and studying them, and it flows very nicely from what we've been discussing. So I'm just going to quickly lay some foundation for how do we build these macromolecules or these biomolecules using these hydrocarbon chains and using the different functional groups. So that idea is macromolecules, how we build life essentially. And first of all, a macromolecule, macro of course means large. So when we join smaller organic molecules together, we're building a macromolecule. And we have four major classes of these macromolecules. We have carbohydrates, lipids, proteins, and nucleic acids. And that's what your lab this week was dealing with and showing you how large some of those chemical molecules are when you start to look at each of the atoms that they're made up of for a single molecule. So when we have a single molecule, we have a monomer. But when we have a number of molecules joined together, we have what we call a polymer. So polymers are long molecules that are built by linking repeating blocks in a chain. So the building blocks of that chain are the monomers, the repeated small units, and they're generally held together with some sort of covalent bond. And the way that they're actually built is through a process called dehydration synthesis. So dehydration actually is a great name for this because what it's happening simply is by joining this bond together, we're releasing a molecule of water. So that's how we dehydrate our polymers as we're making them and putting them together. So we have a hydrogen on one molecule and a hydroxyl on the other. And by having some sort of energy input, we're able to have a bond occur here. These two become a molecule of water, and we have a nice covalent bond replacing it. So let's see. When we're actually doing this dehydration, we can actually, we're taking out water. Okay, and this is actually saying the same thing. And this animates, this slide does. That's why I was hesitating whether I should turn on the animation or not. But essentially, what's happening, this dehydration synthesis is going to require energy in order to build that bond. And in most cases, there's also going to be a need for enzymes to lower the activation energy for this to occur. Another term for this is a condensation reaction because, again, it's the idea of pulling out a molecule of water, and when we pull those out, we're leaving room for this bond to form. Another term for this is synthesis of a polymer, but it's also called polymerization. Now, the opposite is if we're trying to break apart a polymer or digest it. And in order to do that, we have to put the water back in. So this reaction is called a hydrolysis reaction. So hydro being water, lysis being break, or it actually does mean break apart. So by adding water back into this, we can break up a polymer into its smaller pieces. And so we're taking that H2O and giving the hydrogen back and the OH back to the other one. And again, this is going to take some enzymes to break this apart. There is a release of energy as we break it apart in this case. But enzymes were needed again to get it going, to lower that activation energy. And once you do, then you get a release of energy. So this is reverse of the dehydration synthesis. You're cleaving off one monomer